Good afternoon and welcome to the Plant Breeding International Plant Breeding Seminar Series. Today, we have the honor of having a speaker, Dr. Stephen Smith, who's going to talk to us about estimating the global usage of corn belt dent germplasm and the implications for the sustainable use of maize genetic resources. Dr. Smith is originally from England, where he grew up in a farm in Eastern England. And he has a Bachelor of Science from the University of London and a Master's in Conservation of Plant Genetic Resources and a PhD in Evolution of Maize from the University of Birmingham. In 1977, he spent some time here at NC State working with Drs. Major Goodman and Charlie Stuber. And after that, he took a job at Pioneer at the time, Corteva nowadays. He spent 35 years working in Pioneer and DuPont Pioneer, where he received several awards for his work related to intellectual property of germplasm, corn germplasm. He's a member of the American Seed Trade Association, an honorary member right now, and he's affiliated to Iowa State right now as a professor and visiting scientist. In his spare time, Dr. Smith plays the organ and works in various committees related to the Des Moines Symphony Orchestra. And he also serves in the Iowa as a member in the board as a member of the Living History Farms of Iowa. Thank you so much, Dr. Smith, for being with us. And I'm going to be sharing your slides from my computer. So let me put it in presentation mode and welcome and look forward to your presentation.
Well, thank you very much, Carlos, and thanks for setting up this recording. And hello, everyone who was affiliated, is affiliated, or would like to be affiliated to North Carolina State University. Greetings from Iowa. Let's move to my first slide, if we may, please.
Yes, first slide showing. Okay, there you go.
Yeah, lovely, thank you. So maize or corn was domesticated about 8,000 to 10,000 years ago in Oaxaca, Mexico. And from there, during the next several thousand years, spread north and south through the Americas and has been categorized into about 300 to 350 races, including at least 150 discrete types. And I'm relying a lot on the work of Major Goodman and Bill Brown here. So I just want to acknowledge them. Just 6, which is about 2% by number of maize land races, are economically globally important. Those are the Mexican dents, the corn belt dents, which we will be talking about today, the Tucsons, Caribbean flints, northern flints and flowers, and the Catatos or Argentine flints. The corn belt dents are by far of greatest economic performance globally. Okay, thanks. Next slide, please. Well, after the late 1490s and very soon thereafter, actually, maize was transported with other things like tobacco and potatoes into Europe. It arrived in Europe extremely quickly within a decade or 2 or 3 decades. And you can see from this slide how maize then spread from the Americas throughout the world, pretty much. It got to China very, very early. And there were several introductions occurring over the centuries since the 16th century. Many in particular from the late 1800s onwards. Okay, thank you. Next slide, please. So this emphasizes how just important maize is in cultivation around the world. The redder the areas get, the more maize is cultivated. So it's crucial, you know, those transfers happen from the Americas, but here's the result today, that maize is extremely widely grown in temperate and tropical climates around much of the globe. So, and it's the corn belt dents, as we will see, that are contributing a majority of this production of maize. Okay, next slide, please. Thank you. And just to show some numbers here, the US provides 37% or so of global commercial maize production. China, about a quarter. So you add the US and China together, we're up to 62%. Then Brazil next, we're getting to 72%. Argentina, then we're going up to 76% cumulatively. Mexico, we take it to 80. And just with these 8 countries here, we're up over 80% of all world maize production by these few countries. So in this study, which I'll tell you about as through the next slides, we focused on these 8 countries, and then we extended that to the top 15, which collectively covered about 90% of global maize commercial. This is commercial maize production. Okay, next slide, please. Now, this is a slide looking at production of maize through the early 60s till now in those 8 countries. And you will understand, of course, that production, it doesn't tell you what's behind all that production. Was it breeding? Was it weather? Was it more acreage? But here's the production. And in Brazil, let me think, I'm getting mixed up with my colors here. You can see the production has gone up dramatically, 12-fold in China. It's gone up 4-fold in the USA. And you can see the other countries have got less of a rate of production increase. But what's more interesting is in the next slides, I think. This starts to give you some indication of what might be contributing to that increase in production. So China, in that same period, the harvested area has gone up nearly 3-fold. So a lot of China's production is due to increased area of maize production. In US, it's bounced around a bit, but on average, it's gone up 1.5-fold, not too much. Brazil has doubled in the area. Ukraine has gone up 4-fold. But we only have data for Ukraine for relatively recently from the 90s, and that is for political reasons, which we don't need to go into here. Okay, thank you. Next slide, please. Now, this is a bit more interesting because this is looking at yield increases. And you can see US between the 60s and present time, the yield per acre or per hectare, whichever way you look at it, has gone up nearly 3-fold. France has gone up 3-fold. Argentina has gone up 4-fold. China, in terms of yield, 4.5-fold. Brasilia, 4-fold. So there's a lot of increase here that's due to plant breeding, genetics. Okay, thanks. So the next slide. Now, here is an aerial view of North Carolina, and I spent many times walking across the brickyard, one direction to go to have lunch and the other direction to carry punch cards, which many of you won't have even heard of, but they're what drove computers, told them what to do, so we could do our multivariate analysis of isozyme data. So here's Major on the left, and Major took me to my first, and it may have been my last basketball game, which I just, I got neck ache from watching things go from one corner to the other. So, but anyway, Major is a great basketball fan. And Charlie Stuber, for those of you who know him, I mean, Dr. Pepper ought to give him a Nobel Prize for or some award for the numbers of Dr. Peppers that he's consumed. It kept the scoring of PGM and beta-glucosidase going when he might otherwise have been flagging, I suspect. Now, the serious point is this, that this talk is going to talk about genetic diversity, and NC State, with these 2 gentlemen and the students that they've all had with them. Major has focused on the conservation and use of exotic genetic diversity to broaden the germplasm base. Charlie basically was looking at, and perhaps I think was the first to start doing quantitative trait locus analysis using molecular markers. Now, at that time, we had the grand total number of 21. That's not 21,000. That's 21 isozymes. And in US corn, 15 of those were segregating. So there wasn't that much variation, but this was a great breakthrough. And here is some strange person at the bottom being totally mesmerized or confused with the very complicated malate dehydrogenase isozyme. Okay, next slide, please. So this sort of starts bringing it together, because as you increase breeding efficiency using double haploids, for example, and marker-assisted selection, you can see from these slides that you're looking at genetic variants and progress in breeding, and there's various breeding designs there in the different colors. The thing is to notice that the fancy colors, that's not black, they are all breeding schemes using doubled haploids and genomic selection. And you will see, okay, breeding is more efficient, perhaps, but the thing is the variance goes down unless you bring in some new diversity. So, okay, it's important to increase the efficiency of plant breeding, which Charlie Stuber and his students were critical to build the foundations for being able to do that. But if you don't bring in some new genetic diversity, of which Major has devoted his life to doing, and trying to tell everyone that this needs to be done and put some resources into it, that's the other part of the equation. So basically, you can floor the accelerator in a car, but if you don't fill up the gas tank, you're going to come to a grinding halt. So that is a brief summary of how this whole thing relates to sustainability. Okay, next slide, please. And this just shows a graph on the left. You've got genetic resources, and why is it important? Well, you need to fill the fuel tank for genetic resources. And on the right-hand side, this is the simplistic way of looking at how you can make breeding more efficient by identifying genetic regions that are useful. But there's many genetic regions I doubt you'll ever able to be identified. So, but you can identify some, and that helps make breeding more efficient. But you've got to have the germplasm diversity, otherwise you're not going to go anywhere. Okay, next slide, please. So let's look now at how challenging it is, in fact, and has been and still is, to actually measure and monitor genetic diversity in cultivation. So what I mean by that is being able to monitor from year to year in farmers' fields just how genetically diverse the maize germplasm that's being grown is. So this, I think, is what you need to be able to do that. You need to build a database. And being genetically based using marker data is by far the preferable way of doing it. Now, bear in mind that those data are a surrogate, because since we do not understand much of the relationship between SNPs, single nucleotide polymorphisms, and loads of agronomic traits and physiology, you can't go in there and look at the diversity that's affecting yield, because you really don't have much of a clue as to what that is. So you do a surrogate, which is to search and sample the entire genome. Now, that has to be affordable. And clearly, when we were in Raleigh, when I was in Raleigh there, we were doing 21 loci, but and that was affordable. But I mean, it didn't survey the whole genome. And RFLPs, I'm talking sort of ancient technology here. It certainly wasn't ancient when I was in Raleigh. I mean, if we could have gone from 20 to 110 loci, I'm sure we would have done. but the technology hadn't been developed. And it's only now that you've got these really high throughput laboratories with robotics and SNPs on chips that you can actually begin to think about doing assays of several tens of thousands or millions of SNPs across an inbred line or a hybrid. Now, actually, you probably don't need to do anywhere near close to that number, but it's got to be affordable. And it needs to be able to discriminate among inbred lines and hybrids that you really know are different from each other. And you need a database because you've got to monitor change over time. Now, the problem up till now has been this succession of different marker technologies that give you different databases that you can't compare with each other. But now, when you can get down to a single base pair, you can build a database. And that will allow you to compare with global maize, including land races. So you're sort of using one language to read the diversity, although as a surrogate for agronomically useful genes with the technology. And SNP technology, I think, does that now, makes that possible. So why don't you use pedigrees? Well, they're frequently unavailable. They can be wrong. They take no account of selection. They aren't informative on specific chromosomal regions. They just give you a mean kinship value across the whole genome. And often the data, as we will see, are pretty shallow. They only go down a few generations. So molecular markers are the tool you want to use, and SNPs seem to be pretty much ideal. And they haven't been available until fairly recently. There's a lot more problems, though, coming up. Okay, let's just go back and look at what the corn belt dents are. Well, firstly, if you see corn, hybrid corn growing anywhere in the USA, it's basically corn belt dent. Okay, and that originated in the US during the 17 and 1800s by hybrids between a couple of races. The Northern Flints, which had been established in New England by 1250 AD, if not before, actually, when, if you know your English history, William of Normandy invaded England in 1066, and corn was being grown in what we call New England at that time. Of course, William the Conqueror and the English didn't know about the American continent. But that gives you some time scale. So maize was in the US, well, North America, we should say, and it has been in North America for the last 3,000 years or so. But the Southern Dents actually is another race that only got into the southeast of the North American continent fairly recently, in the last during the last 400 years. And they have a very broad, diverse genetic background, which really is probably not well understood and really needs to be, because the Southern Dents mixed with the Northern Flints, but most of the corn belt dent is probably Southern Dent germplasm. So here's a problem. We don't know a whole lot about the basic origins and diversity in the corn belt dents. Now, I'm sorry, but that is ludicrous, because this is the primary genetic base of US corn. So there's a big research project there. Now, there is some what we might call exotic germplasm in corn belt dents, and Major has looked at this quite a bit, and it's awfully difficult to find out just how much there is and where it is, because breeding organizations might say they put some exotic in, but exactly what they mean and is it in their most elite material is another other issues that sort of they might not want to talk about too much. So there is some exotic germplasm and in a few hybrids. Maize Amago from Argentina is one of the most important. And what you find then is, of course, that then it sort of gets incorporated, and then it's all part of corn belt dent, and then it goes global. So bear that in mind. Okay, next slide, please. So, this seminar is telling you nothing new, because CBD germplasm usage on a global scale has been well established by a whole load of people. some of whom are listed here. It's well adapted to temperate environments. It is used in the subtropics, and it is used by CIMMYT, the Wheat and Maize Improvement Center in Mexico, for improving yields in semi-tropicals and tropicals. And it is increasingly used by commercial organizations in tropical environments. So CBD is well adapted to temperate, but it's also, and it's, as we will see, it's used a lot in around the world in temperate conditions, but it's also being introgressed into tropical environments, which is good to improve breeding. But then you've got to start wondering about, well, okay, isn't the germplasm base getting a little bit too much the same everywhere? That's what you need to be thinking about. Next slide, please. So I mentioned pedigree data isn't the best to use, but let's just have a look at it, because if you do have pedigree data, it can be somewhat interesting, particularly when it's not correct. But that's another point. So pedigree data on inbred parents of hybrids is usually maintained as confidential, so it's not publicly known for most hybrids. The pedigrees of inbred lines are known via utility patents, but the depth of information is not very great. So you can't usually track much to publicly known founders. And the relative usage of inbred lines as parents of hybrids is confidential and not disclosed publicly. Now, there was a time up till the 80s when publicly developed inbred lines were widely used in the corn industry, and the American Seed Trade Association did studies, surveys every 5 years to understand usage in those lines and change in usage. And that was very useful, actually. But as soon as those lines were parents of other lines that were then proprietary, or some other germplasm came in that wasn't particularly represented in those public lines, then the pedigree information really wasn't very useful. And if you don't know which inbred is used in which hybrid, and you don't know the relative usage of the hybrids, I mean, I'm sorry, but that information isn't particularly helpful in terms of monitoring diversity. So that's why it gets back to the fact that marker technology is the way to go. But there have been and there remain big problems to actually getting molecular marker data. So as I mentioned before, it really is now enabled technically. It's enabled technically by this 25-year evolution up to get to SNPs. But okay, you've got the technology, but then can you use it? Well, the last survey of diversity of widely used US hybrids was published by me and co-workers in 1992 using RFLPs. Now, you're wondering, you should be, why on earth have there not been any more studies since then? Well, if you go and have a look at the bag tag licensing language on hybrids, you will see that it basically prohibits marker profiling for commercial and perhaps for any purposes. And you know, if you're doing research in a commercial breeding organization and you're going to publish it all, it gets to be awfully difficult to persuade lawyers and such that you are not engaged with commercial activities, even though you're publishing. The use of different GMO stacks based on the same genetic foundation complicates sampling, but I think a sampling could be worked out. That's one of the two big things are, how are you going to sample the diversity so it's meaningful? And then are you legally able to do it? And then getting the resources to do it, I think, is probably, unfortunately, the least of the problems. If you want to start marker profiling the parental inbred lines, bearing in mind you don't know which hybrids they've been involved in, you have to wait 20 years until the patent runs out before you can do that. And as I said, the relative usage of those inbreds is confidential and no one knows. So really what you need to do is survey hybrids or the F2 seed from hybrids that is currently grown you've got to work out a sampling strategy that does a proper job, and then you've got to have legal freedom to do the job. And those are the two, perhaps the legal ability to do the job is the most challenging. But that's why things, at least in the US, have ground to a halt in this area. Okay, next slide, please. Now, you can get some idea of how germplasm has changed. This is looking at SNP-based diversity for inbred lines from inbred lines that were plant variety protected, and then after 20 years, they're in the public domain. So they can be profiled then. And I'll just let you, at your leisure, I'm sure you can hit the pause button, look and see. So on the left, we've got, and these were the available data from White Crop Science 2020 actually, 2019's electronic publication date. So you can compare Holden's and Pioneer for different germplasm backgrounds. In the inbred lines, they PVP'd, and they PVP'd those one between 80 and 84. And then between 85 and 89, there's 4 different companies you can look at. And then between 90 and 95, there's 5 companies, and you can see the difference in the germplasm backgrounds between companies and in time. But just what does this mean in terms of what's out there in hybrids? Well, I'm sorry, but I really haven't got much of a clue, other than you could assign some of these backgrounds to males and some to females, and you're going to need 50% of males and 50% of females. But then you don't know which hybrids were the most widely grown. So, you know, these data tell you something, but they don't tell you a whole lot, and they're 20 years too late at best. Okay, next slide, please. Thank you. So this is the study from 1992 using RFLPs, and it's a dendrogram, and it's portrayed this way, like a mobile hanging from the ceiling. And actually, if you look at this, you can see on the bottom, if you go 95B to the right, you've basically got one big cluster, and then to the left, you've got another big cluster. And then within the right-hand side, you've got several groups of hybrids that are pretty similar to each other. They join together, you know, not far into the distance. And so this looks like pretty much the same inbreds being used by different companies, at least on the far right for sure, and some in the middle there. And on the left, then those hybrids are much more different from each other and from others. And that was the picture in 1992, but from the harvest of 90 and 91. And these hybrids were picked on relative usage from farmer surveys. That is the last picture we have of diversity in US maize. Now, I think that's quite appalling that we don't know anything more between then and now. I think that's a major concern. Okay, next slide, please. Now let's, we're going on a trip to China, and we just got there. Now, what's the situation with pedigree data in China? Well, it's different. This is a different situation to the US. So the inbred parents of many or most hybrids is actually publicly disclosed. Now, whether that's going to continue in the future, goodness only knows. The pedigrees of most inbred lines are publicly known, but again, the depth of information isn't that great. Back to the founders. The relative usage of hybrids, the most widely used hybrids are publicly reported. I haven't yet found, though there may be in Chinese, systematic surveys of diversity. Let me just backtrack to say the depth of information. There's a lot of usage, or there was a lot of usage in China. Single cross hybrid started in China in the 60s. And there was a lot of usage for the first 3 decades of US inbred lines: Missouri 17, C103, B73, Ohio 43. So, I mean, the pedigree data track back to those, and that's nearly as far as you're going to get, basically. Now, with marker technology, as I've said, this is enabled by SNPs. There are many published studies of SNP data on Chinese inbred lines, 5, 6, or 7 of them, using SNPs, some with simple sequence repeats. And many of these studies actually do analyses, a structure analysis that puts the inbred lines according to this population structure into various groups, like stiff stork, iodent, or some Chinese land races. So we'll look at those data coming up. But again, if and when China has GMO stacks on the same basic genetic foundation, that complicates, well, what's a different hybrid? Well, the base may be the same, but if it's got a stack on that, how am I going to characterize that? Is it a different hybrid or not? And so I don't know what the situation will be in the future. Will it still be possible to just obtain inbred lines and profile them with SNPs? Because there's likely to be more proprietary inbred lines in China. That may be a block. We don't know. Next slide, please. And this gives you an idea, and it's just an idea because you don't need to look at the details here, of what Chinese maize germplasm looks like. And you can see, if you have heard anything about US corn, there's some names here you're going to recognize. NSS is non-stiff stalk, SS is stiff stalk, IDT is iodent. Mo17 is the old famous Missouri 17. B73 is B73, and etc. And there are some Chinese groups here from land races, and China developed land races several hundred years ago. So, for example, we have the Luda Honggu group here mentioned on the left, and the Huangzaozi Tangxi Pingtao group in my best Chinese. So what we're going to look at in the next few slides is, excuse me, just how much of the US and how much of the Chinese germplasm is in the hybrids that China cultivates. Next slide, please. And so you can, there are several of these studies I mentioned, but I used here the study by Wu et al. In 2015. And they looked at a bunch of, well, several hundred, 1,000 perhaps, Chinese inbred lines. And they did it with SNPs, and they did structure analysis, and they found, or they organized the germplasm base into these 7 structure groups. And you can see the US stuff is B73, 207 is iodent, Ohio 43, Missouri 17, PA, gosh, the P stands for pioneer. That's germplasm developed from a pioneer hybrid. And then we have the Chinese germplasm, Hangzhouzi and Tingxing Pingtao in green and dark blue. And here's just the snapshots from the 60s and going to the 2000s. And you can see a change in the germplasm backgrounds. Now, these data are not weighted according to relative usage because we really don't know enough about that. But there are 2 hybrids that are grown over about, and they're derivatives at the moment, that are grown over about 80% of Chinese maize agriculture. And they contain a high proportion of corn belt dent germplasm. And you can see in any case, there's a lot of corn belt germplasm here in China. And bear in mind, China is the 2nd biggest producer of commercial maize in the world. Next slide, please. And this shows the change in time in China going from the 60s now through to the 2000s. Excuse me, I'll take a quick drink here. And along the bottom, there's B73. Its use wasn't huge. It went down, it went up again, and it's starting to go up again a little bit. The orange is iodent, and a fair amount of iodent in Chinese germplasm. Ohio 43, a heck of a lot of that. It's decreasing a bit. A big reliance, even till now, on Missouri 17. And I mean, B73 and Ohio 43 and Missouri 17 are, I don't know, 40, 50 years old. I mean, I'm older than that, but only just. And then PA, that was from a Pioneer hybrid, and it has really taken off from the 80s into the 90s and is continuing. And then you see the germplasm from the Chinese land races in the dark blue and the green. And well, they increased in the 70s, and then it's sort of decreasing somewhat. And so you can see that the US corn belt dent germplasm in China is contributing, well, around 80% of the germplasm. That's not weighted by usage, but if you look at those two hybrids that are the most widely used in their progeny, that's about the number. Okay, next slide, please. And okay, this is the only publication I found that actually gives you some indication of how things are changing in terms of planted area. But it tells much the same story, because the Chinese germplasm, the Tang Xingping Tao and the one at the bottom there, Luda hybrids, inbred lines, are in the minority compared to the US germplasm. So when you can actually see some indication of percent usage by acreage, the corn belt germplasm is very highly significant in China. Okay, next slide. Now, when you start trying to find out information from other countries, you might think the available data from the US is not good, if at all available. China has got more data available, but we don't know what will happen in the future. I could find much less published data on this issue for other countries. And what I could find, I mean, this is a brief summary here. So Brazil is primarily tropical, but corn belt dent stiff stalks were introduced in the 90s. Argentina has got a lot now of corn belt dent germplasm in it. Europe has a heck of a lot. But, there are hybrids in the Midwest that will grow just fine in North Italy, for example. South Africa, US germplasm went in there in the 50s, and now it's very high proportion. And you see something similar happening around the world as you happened in the US, and that is that iodent germplasm that was initially used primarily and only by Pioneer, really, then has been much more widely used in the last 20 years by other breeding companies, and iodent is being much more widely used in other countries, other temperate regions of the world. So in terms of, so those are pedigree data. Marker data, there is some marker data available showing that there's corn belt dent germplasm in Brazil, so it sort of backs up, as you'd expect it ought to do if the pedigree data are correct. It backs up what you have been looking at here from the top half of the slide from pedigree information. But there's no systematic studies at all using marker data of diversity changes in time, basically anywhere. The most detailed study I saw was for Cuba about 30 years ago. And the European Union actually did a study about diversity change in a whole load of crops about 30 years ago, but I've seen nothing since. Okay, next slide, please. So when we looked at these data, myself and Alan McCunn at Corteva and Wally Huffman, who's an ag economist, he's sadly passed away at Iowa State, we were looking at this. This is where we sort of got to, and it really is only an estimation. because that's all you can do with the current available data, which, as I've said, is I think, a major concern. So let's say the USA is 100% Corn Belt Dent. I mean, let's assume that Mesa Mago now has been incorporated, and basically we can acknowledge its origin, but decades ago, but it's a package that goes with Corn Belt Dents now, and it'll probably go around the world, even beyond Argentina. So US maize production is 36.6% of global maize production. So that's all CBD. We estimate China of 80% Corn Belt Dent. So that means that 23.1% of global production coming out of China is Corn Belt Dent, based on Corn Belt Dent. So you can see how we did these calculations, and you can disagree with the numbers on the left somewhat, but if you do, it would be great to see the data, because then we can improve this study. I don't know if I can call it a study, because it's based on it's sort of looking into a crystal ball to some extent. But basically, you can see that around 80% of global maize production is dependent upon Corn Belt Dent, and this is worrying, because if you're worried about, and you should be, about over-reliance on one particular germplasm base, this should ring some alarm bells. Okay, next slide, please. And there are some comments that back up this, this high usage of Corn Belt Dents. Use of temperate maize germplasm in tropical breeding programs has been poorly documented, but in general has been growing. Many tropical hybrids contain some temperate germplasm. Among other introductions into India, stiff stalk and Lancaster germplasm were used directly as commercial cultivars. And for one international commercial breeding organization, the similarity of hybrids to US temperate germplasm was estimated to have increased by 10 to 12% during the past 4 decades for hybrids in Brazil, Mexico, and India. Next slide, please. Now, let's look at bringing some diversity into the US. And there are several programs that have been working to help achieve this. One is the Germplasm Enhancement of Maize Project, that is coordinated out of the PI, Plant Introduction Station here at Ames. The North Carolina State University breeding program is a critical part of that. You have Major's NC State program that adopts tropical inbred lines to temperate environments. You've got a North Dakota program that brings in some exotic germplasm. So these programs have demonstrated the positive potential of exotic germplasm to provide new useful genetic diversity. But it needs more work with that germplasm to get the useful bits of it into temperate maize germplasm. Okay, next slide, please. Now, this is from White, Crop Science 2020. When you look at, and they were looking at pedigree backgrounds too, they also have marker data in their supplementary materials, but they were looking at pedigree data here, trying to understand the backgrounds of the off-PVP inbred lines, or actually inbred lines that were still under PVP. It's the marker profiling you can't do until they're off PVP or off patent. And you can see, and this is a problem if you're going to use pedigree data, but as I've said, this is the least of the problems, because you've no idea which hybrids are using which inbreds. And the gray is getting bigger and bigger, and the gray is unknown. And that is because it's being increasingly difficult to track the parents to parents that you know anything about, because you can relate to them as public inbred lines, for example. So pedigree information is getting, well, it's not much information at all, actually. And White and his colleagues conclude that it's likely that a majority of the unknown pedigree is in fact derived from within the primary historical breeding pools, although integration of new alleles has been emphasized by breeders within these companies. Okay. As I've said earlier, just what is meant by exotic germplasm and how much of it and in which hybrids is a bit of an unknown, at least outside those companies. Okay, next slide, please. And what's happening to germplasm diversity in the Corn Belt dents? Well, this is a figure from a study 2012 by Van Heerwaarden et al. And you have inbred lines from three eras, the 50s, the 80s, the 2000s. And they looked at stiff stalks, non-stiff stalks, and iodens. So in stiff stalks, for example, the reddish type things, the effective number of direct ancestor haplotypes went, well, it went down dramatically from the 50s to the 80s, and then sort of went up a little bit in the 50s, went up a little bit. You look at non-stiff stalk, wow, what a decline in diversity between the 50s and 2000s. And iodent is sort of going down. And if you very simplistically just do a linear regression on those data, this is when the germplasm will run out. And it'll actually run out earlier than this, probably, if you don't bring anything new in, because this wasn't taking into account use of double haploids and genomic selection. So basically, non-stiff stalks, by this estimate, ran out already. They may not have done, but that tells you the trend and the concern things are going. Iodents have got a bit more life in them, according to this simplistic, and it's probably completely oversimplistic way of looking at things. Stiff stalks, they don't have a whole lot left in them, do they? And if you get down to one heterotic group, well, can you make a hybrid? Well, probably not. So, I mean, people should take this into account. And Major has been trying to get people at breeders to take this into account for decades. And they really better. They better, because if you leave it too late, you leave it too late. Okay, next slide, please. So, you know, there's much hype and focus on using identified single genes, whether that's mapping native germplasm, using markers and finding quantitative traits and mapping those specific traits in the germplasm, or finding genes in other organisms and engineering them into inbreds and hybrids. So there's a whole lot of focus there. And because one reason is it's really glitzy, and the stock market really can get turned on by this. But there is far less overall focus or support for the development of a broader genetic base and its continued improvement. And, you know, Major and the people working with him, and really because of him, have put a lot of effort into this and are still doing so. And it doesn't require a huge technical experiments and huge, you know, it's field work. It isn't glitzy. But by golly, it's crucial, because, you know, just how many genes can you identify that are going to significantly increase yield? And let's face it, BT is basically just turning the corn plant into an industrial facility for producing BT toxin. I mean, so instead of having an industrial plant producing BT, you're producing in the plant. Now that is advantageous, because then it's inside the plant. So these little borers who hide away from pesticides will then be exposed to BT. But the point is that, you know, both these approaches, the single gene approach and the multigenic anonymous gene approach, And, you know, genomic selection is a marker-assisted way of dealing with a lot of anonymous genes that affect multigenic traits. So both these approaches, bringing in some new germplasm, really exploring it, trying it out, and trying to find a few silver bullets, of which there are probably none, let's face it, for quantitative traits. Both approaches, they are really complementary, and they provide not only new genetic resources, but also fresh opportunities to increased understanding of how these complex interactions with genetics and physiology and agronomic practices work. So, I mean, with the single gene approach, there's a lot of hype with gene editing. But if you don't know the traits you're interested in, and then even if you do, if you don't know the genes you're interested in, I mean, what are you going to do? And these things need to, I mean, it's just common sense, really. It's all nice hype, and it's certainly got a use, but it's got a use for certain things and not for others. And we need to make sure the resources are applied to the areas where they're actually going to have some impact. That might be the last slide. We'll soon find out when you press the button. Okay, right. Well, thank you very much for bearing with me through this presentation. If you have any questions, you can get hold of me at jscsmith@iastate.edu. Thank you very much.
Thank you, Dr. Stephen Smith. Very interesting presentation. And yes, the audience can guide the questions directly to you, or they can send it to me, and I forward it to you, Dr. Stephen. Thank you so much for your talk. It just comes to mind, we both have worked in seed industry before. And before I left Syngenta, I could see that breeders and geneticists were incorporating some measurements of genetic diversity into their predictions. So it was not everything a lost hope, in a sense, that we're just crossing good by good and narrowing the genetic base. A lot of the modeling that is being done takes into consideration what is going to be the ability to sustain the genetic gains into the future. It's not just to maximize the current genetic gains. So hopefully these companies don't run out of gas. As you said at the beginning, they have a very good car, but they run out of gas because they finish with they end their genetic diversity. But that's a hope. Okay. Thank you again. Thanks, people, for watching this recording. We have, let me show this. As I said, the questions can be forwarded to me, and I will forward to Dr. Smith, or I can, when we share this recording, we can post Dr. Smith's email directly for people to forward questions or comments directly to him. Thanks, Brandon, and for your help in this seminar, and also looking at the next one, which is coming up on Thursday, October 21st. We're going to have Dr. Motoyuki Ashikari from Nagoya University, who's going to talk to us about rice breeding for Africa using useful agricultural traits. He's actually going to be speaking to us at midnight Japan time. So I hope you understand the circumstances. It's great that we have a person from Japan presenting a seminar for this international series. Thank you so much. You have a great week ahead, and look forward to seeing you back in other seminars. Thank you so much. Okay. Stop.